And so what we want to do is implement what a computer scientist would recognize as an abstraction hierarchy supporting programming in DNA. So we'll just make this up and see how it goes. Parts are basic biological stuff, and we'll hide the DNA, and we'll give it a number, and we'll give it some functional information so you could use it without having to know all the biology. And then we'll build devices out of parts. These are just a bunch of numbers. These 4 parts come together to make an inverter or a NOT gate, Boolean logic. This turns out to be a bad design because the input and output signals aren't the same. So we reorganize the black box and we make a common signal carrier inverter and blah, 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 blah. Then you put the inverters together to make a ring oscillator, and we're bootstrapping up an abstraction hierarchy. If that didn't make any sense, you can go to the web and get this comic strip which explains how to implement abstraction for genetic engineering. Okay, so as a result, we now have the beginnings of an abstraction hierarchy. It's incredibly primitive. But practically what it lets you do is somebody gets to become a systems engineer, the other person at the bottom becomes an expert DNA constructor. The person at the top doesn't need to know that DNA is made up of 4 bases, but you still get to engineer a biological system. In doing all of this, we're stealing lessons from all other forms of engineering. So mechanical engineering from the 1860s, where in the US, thank God, we finally got standardization of screw threads, 60-degree angles squared off at the top, so that things snap together and work when you put them together, so long as you don't mess up the English metric stuff. We started a registry of standard biological parts. These are open source genetic objects, parts.mit.edu. There are about 2,000 different parts in the catalog right now. They stink. They're not very well characterized. They haven't been refined to support reliable functional composition, but they will snap together and you can see what happens. Here's our best part. We've got a data sheet, for example. This is a receiver, a genetic receiver. You put this in a cell, and the cell stands ready to respond to a small modified sugar or a signal that comes in from the environment. And so this is the architecture of this data sheet is lifted from the TTL data book from Texas Instruments. If you're familiar with that, you got the transfer function, the response time, the specificity of the receiver to different input signals. The one thing that's different is the bottom right plot, which is the evolutionary stability of this device. How quickly will it mutate as you go from one generation to the next? Yeah. What's the license and so on. So one of my projects for now is starting a factory that makes lots of parts and puts them in the public domain. So it's a lot of work to do that. And so to sort of transition to the last part of the talk, as we've been implementing these very naive and primitive lessons from other forms of engineering in support of genetic engineering, what we're finding is people like to make stuff. So here are students going back to 2003, 2004, 2005, 2006. These are students who participate in something called iGEM. iGEM is the International Genetically Engineered Machines Competition, which is a worldwide genetic engineering competition for undergraduates, in which teams of students from around the world compete over the summer to design and build a genetic engineered machine of their choosing, so long as it's a constructive thing. And so here's the world map of the teams from 2006. And you'll see we didn't have a lot of teams from China, so then we went out to China and we got 5 schools from China to come in. And so here are the students from this year. And so, for example, the team from Melbourne, Australia showed up with some new BioBrick parts. They wanted to change how bacteria float, give you that option. If you're working on some project, I don't know what, but you want bacteria to float or sink or remain neutral, well, here's the part for you. This piece of DNA encodes a little protein balloon that is somehow gas impermeable, and the protein balloon boots up inside cells, and depending on the number of balloons that you boot up, you'll change the buoyancy of the cells, right? I didn't know anything about this biology. They showed up, they refined it, they made it compatible with what else we've got, so you're free to use it. There are probably tens of thousands of other things like this that we need. And if you wanted to help make some of those, that would be terrific. So this question from before, reprogram E. coli to smell nice, was one of the student projects. Here's the team that did it. They're 5 teenagers, and in a period of 4 months, they implemented a 24-component system. And this looks, maybe a little bit detailed, but note that there's no DNA sequence on this system diagram, right? There's things like BSGD, which is a banana smell generating device, right? And so they were able to add in a bunch of parts into E. coli, remove some of the components that make E. coli smell less than good, and try it out. So they tried it out we have a jamboree at the end of the competition where everybody shows up, and their results of their smell test are shown here. They have the natural bacteria, which almost 90% of the people agree is stinky, and then the mint is kind of hard to smell, and then the bananas is pretty powerful. Okay, so this is sort of where we are, right? This is the high watermark of genetic hacking. So let me end in the last 5 minutes with some questions and a briefing on what you might call social issues, or we call issues of human practice. I probably spend 60 to 80% of my time on the questions in this slide as opposed to the stuff in the lab. So, yes or no? Biosafety level 4 is sort of hemorrhagic fevers for which there's no cure and they're transmitted as droplets. So should there be top secret labs working on these things? Will biohackers be good or bad? Is garage biotech inevitable? Should the parts be patented or freely available? Should genetic engineers sign their work? Lots of questions that come up around safety and security and what have you. And what surprised me is how exciting the technology of biology is to people in the hacker community. So here's Make Magazine showing you how to make a gel electrophoresis system for separating DNA out of Legos and a 9-volt battery daisy chain. Here's a thermocycler for doing polymerase chain reaction to amplify up DNA. Here's a clean box for working on clonal propagation of spores by reverse, just decompiling and reassembling an air filter. Pretty sophisticated stuff. And then, of course, there's an impressive article, a genuinely impressive article in 2600 from a number of years ago that I was accused of writing, but I didn't, unfortunately. I mean, it's really well written. And this is my first experience with this hacker community. So DISA, Unix Security Reality, Living Without a Social Security Number, Hacking the Genome, written by Professor L anonymously. Beautiful writing, teaches you how to clone a luciferase gene that makes light into E. coli, and then if you wanted to do this with your dog, go take a course. But the motivation's spot on. Biotechnology is so unbelievably cool, and the industries that we've got thus far are not realizing the potential of the material. Let's get more people doing it. So about 30 years ago, when recombinant DNA technology was invented, people freaked out. Simplest way to say it. So in Cambridge, Massachusetts, where I'm from, using recombinant DNA technology was outlawed by the city. Not allowed to do it. And in response to that, the scientific community had a big conversation about how to practice recombinant DNA technology safely. There's a famous meeting covered by Rolling Stone magazine in Asilomar, California in 1975. And basically what they figured out how to do, and I'll get to this in a second, is move the new technologies of recombinant DNA into the existing recommendations for biological safety. Now, at the same time that this was happening, other people, this is the Subway newspaper from Boston in 1977, 30 years ago, where there's instructions for how to clone a toxin gene in your kitchen. Now, what's interesting to me is I don't have any reports of this actually happening. And I don't know it's because nobody really wanted to do this, or people tried to do it and they killed themselves. And so there was a strong negative selection against hacking stupid stuff. So there's biosafety levels and whatnot that you can learn about. There's different, dangers to the researcher, the environment, or the public that you want to be aware of if you're thinking about this. In the US, if we get money from the government, the National Institutes of Health at MIT, and I'm doing DNA engineering, I have to go through a CAB, the Committee on the Assessment of Biohazards, which includes members of the public, and they review the work before it gets done. There's problems with this system, but there's frameworks like this that I get to inherit 30 years later. This project I described in France, where they remade a virus from 5 million years ago, if you read the last paragraph of the paper, they talk about the safety precautions and how they won't ship you the virus unless you pledge that you will only do work on it under biosafety level 3. So we've got really strong biosafety level frameworks at institutions. But what happens when it becomes easier and easier and easier and easier to do the engineering work, and people do that themselves. So what's changed since the 70s? Well, we've got databases with sequence information. I showed you the phage sequence. We've got the internet. We've got DNA synthesis technology. We've got FedEx and overnight shipping. And in the US especially, and maybe we're just freaks, but we did have an anthrax attack in the fall of 2001 on the Senate. We have concern about people misapplying biotechnology. So if we go back to this website, let me show you one more thing. You could type in a different thing up here, and you would find different DNA sequences. For example, this. And if you went and got that DNA sequence, you would have the DNA sequence for a hemorrhagic fever, which is normally hard to get because you'd have to go to the next outbreak, risk death, collect the sample, right? But because I can get this sequence online, and because they have DNA synthesizers which can convert this information to material, right, all of a sudden the chain of control around some of the dangerous human pathogens one might be concerned about is circumvented. And again, if this is the world today and building this sequence of DNA would cost you about $20,000, in 5 years so that's about a Volkswagen. In 5 years it'll probably cost you about $2,000, right? And then it becomes cheaper still. So that's the world that we're in now and heading towards. I'll skip over this. So I was wondering whether I should come talk about this stuff, and the security issues I felt okay about, because I had an odd experience, a good experience. I wrote a paper with the heads of a bunch of DNA synthesis companies and a bunch of folks from the FBI. And the purpose of this paper was to start a framework for screening all of the orders going into DNA synthesizers at companies, so that the companies wouldn't unknowingly construct human pathogens and give them out to people. And I'm sure somebody here could figure out how to circumvent it, and I hope that if you could figure that out, you would let us know. Last thing, then, in terms of social issues, I won't cover them all. Come back to this project. This is 24 different genetic parts. If you look at how genetic parts are treated as objects today in the legal system, they're treated as patented objects. 3 minutes. So here, for example, is a patent covering uses of green fluorescent protein. This is a protein that comes from a jellyfish, and when you put it into other organisms, bacteria or mice, they glow green. So you can think of this as a print statement, right, for reading out what's going on. So this is patented. Here are engineered proteins that bind DNA. You could use this to turn things on and off, like if-then. This is patented. And then here's amplifiers and latches and flip-flops. And what we see playing out right now is that all the low-level functions in genetic engineering are being balkanized and owned by different people. And so you could imagine trying to write a piece of computer code where if you had to write this program, you know, the person to your left owned the define, and the person to your right owned if, and I owned the left parentheses, you know, and so on. And every time you wanted to write a new code, you had to go get permission from each of these folks, and that wouldn't scale. And so this problem gets worse as we make bigger and bigger systems, and as the cost of making them becomes cheaper because we can print the DNA from scratch, and we go to VLSI genetics, if you will. And so right now we're here, and we're thinking about moving to copyright, but the lawyers are freaked out about using copyright because it seems to never go away, and I heard that the Egyptians were going to copyright the pyramids. So if we mess up transitioning to copyright for genetic sequences, that could have big costs. Contracts leak, so nobody wants to use them. Public domain could be hacked around by people layering patents on top. And we could do something new specific to genetic engineering, but the politics of that are very dangerous and expensive. So this is an area of active work. If you're interested in this specifically, there's a not-for-profit I'm part of called the BioBricks Foundation that's trying to figure out how to solve the legal hack. So last slide. Sui generis. So I guess it's Latin. I won't remember it, but it means specific to the domain or something like that. I'm not a lawyer. So the big examples in the U.S. would be ship hulls, right? So when people were doing competitive yacht racing, you know, you could protect the shape of your ship hull, and there's a special legal structure to protect ownership of ship hull shapes. The other good example is MaskWorks, which people here might be more familiar with. In integrated electronics design, you have the masks that are used in the fab process, and you can't make perfect photographic copies of those, if I remember correctly. So maybe we could do something, we could just declare that this is so new, we got to do something new. Okay, so will there be biological hackers? I think there are.